Okay, I'm gonna go off camera and on mute.
Right. Can you all see my screen? I'm assuming so, yes. Okay. So I will go through these slides, and Amber, if I mess up, you can just chime in. You can unmute and chime in. I'll just wait just a couple more minutes to make sure. Anybody else who is signing on? All right, I guess we'll go ahead and get started and welcome everybody to our virtual lab meeting. Let's see, first up is Bo's podcast, Stronger Every Day. He releases it every Wednesday, so make sure to check that out. You can find it, I think it's on Spotify. I know we have a link on our website. If there's anybody new here, now is a great time to go ahead and introduce yourself.
I am going to unmute and introduce myself. My name is Oleg. I'm here at the Netherlands Cancer Institute, and my main project is on USB7.
Wonderful. It's nice to meet you.
Nice to meet you as well.
And I guess I didn't introduce myself. I'm Becky, and I'm with the Foundation for Hao-Fountain Syndrome. And my daughter Ella was diagnosed at age 4. And I met Bo the same day she was diagnosed. We met over Facebook. I was able to find him. So that was way back in, I think, 2016. Anybody else new? I think I see a few names.
Yes. Hi, everyone. Here.
Hi.
Hi, my name is Fatemeh Rafi-Nejad. I'm an occupational therapist, and my last published work was on about a case report about a patient with Hao-Fountain syndrome from Iran.
Well, welcome. We're so glad to have you
Thank you.
here.
Thank you so much.
Right. Is there anybody else? I think that is everybody that I think that's everybody that might be new. There is some new research, and we are so lucky to have a couple new papers that are out. This one here, the small molecule allosteric activator. And then we also have this one as well, activation dynamics of ubiquitin specific. It's hiding on my thing. Protease 7, I think it says. I can't. It's hidden on there. Oh, there's a rare watch party, and I know Amber is going to send out a link for this for Friday, November 28th for this rare documentary, and she and her son are featured in this. So she's going to put a link in a follow-up message as well. These are some of our resources. We have the full biobank. We have a resource spreadsheet. And then we are collecting data on Matrix. This is all of our patients. We're trying to get everybody on here to join, and there will be a lot of information on Matrix that researchers can get from the Matrix. And then we'll start here talking about Nashville. We had such a great time at our conference. Just it was a great 2 days. The first day was focused on scientific. Day 2 was focused on patients. I think that I think Tara and Dennis are going to go over day 1, right? Yeah. Okay. Just making sure that's correct. For day 2, focus on the family. We really talked a lot with Christian about their information that they found from their phenotyping study, which was really good information about the patients and the different symptoms that are most common so that we can start to build on that. And hopefully we can incorporate Matrix in with their data to get a really big picture in the next year over what it looks like for patients. And we heard from families about their stories and their experiences with different symptoms. But it was really great 2 days, and I'm so glad that for everybody who was able to make it, and hopefully next year everybody and even more can make it. It was a really, really fantastic time. And let's see. No, I'm gonna go ahead and hand over control now to Dennis and Tara. Let's make sure, let's see, are you guys already set as hosts? And let me stop sharing my screen. Let's see, are you guys able to share?
Yeah, I think
Yes.
so.
Okay, perfect.
Okay. Do you see it? Do you see our screen? Perfect.
Yep.
Well, great. Thank you, Becky. Good morning, everyone. My name is Tara Bayat, and today with Denis Ishtapihar from Fantasar Lab, we are giving you a quick recap of the recent Hao-Fountain syndrome symposium. And as Becky said, it was in both a scientific session and a family conference, and it was a pleasure attending this meeting for the second time. And as you may know, it was this year, it was in Nashville, Tennessee, September 18th to 20th. And we attended for the scientific session on September 18th, which was amazing. And I think we are growing. We had more attendees this year compared to last year in Atlanta. And so the meeting, maybe I can just, good. So the meeting began with Bo and telling us a very inspiring story about how hard it is to find a doctor for Hao-Fountain syndrome. And knowing Bo, I know he didn't give up in this way, and this is why we are all here today. And Becky mentioned Bo's podcast, and I wanted to bring it to your attention again, A Stronger Every Day. And this is an amazing podcast by Bo, and I try to listen to one episode at least every day when I drive to work. I try my best. And Becky gave us a very positive updates, very amazing news on ICD-10 code for Hao-Fountain syndrome, which is effective from October 1st. And also she talked about the need for a clinical center of excellence. And also she mentioned that the majority of Hao-Fountain patients are located in East Texas and Florida, if I'm not mistaken. And also Mandan, who is the father of a Hao-Fountain kid, talked about his personal experience of living with a Hao-Fountain patient. And this is a very a unique story, a very unique experience. And it was amazing listening to his talk. Yeah, and then we moved to the presentations, and we started with the first presentation. It was Tyler. He's a PhD student in Dr. Larry Reiter lab at Tulane University, and he gave a talk on modulating circadian rhythm through USB7 targeting small molecules. And in their lab, they're experts in circadian rhythm, and Dr. Reiter has been working on circadian rhythm in Prader-Willi and Hao-Fountain syndromes, and they've been working on Hao-Fountain recently. And they developed a model from dental pulp stem cells, and also they encouraged more Hao-Fountain patients to participate in their studies, and they can send them a kit if they're willing to participate in their study. And I just wanted to highlight this, maybe I can. Yeah, I just wanted to highlight one of their recently published paper. It was a few months ago. And if you would like to know more about the mechanism of the circadian rhythm. And next, we had Dr. Boris Bonaventure. And Boris, please correct me if I'm pronouncing your last name wrong.
It's perfect. It's perfect. Thanks.
Good. And he gave a very interesting talk titled "The USB7 TRIM27 complex restricts flavivirus infection by limiting virus entry into host cells." And he's an expert in mass spectrometry, and he brought these questions in his presentations. How do viruses affect cellular processes? How do human cells respond to viral infections? And what is the function of USB7 in virus immunity? And then he moved to the USB7 and to his findings on USB7 mutations and the infection and how it affects the virus entry. And the next speaker was Lieselot van der Loon from Amsterdam University Medical Center. And she gave a talk on DNA methylation episignature and comparative epigenomic profiling of Hao-Fountain syndrome. So they had almost a small cohort of patients. I think, if I'm not mistaken, 22 patients with Hao-Fountain syndrome. They investigated their epigenetic signature, and it was quite different between patient to patient. However, this brought us to this need for a larger cohort and also for encouraging more families and patients to participate in this interesting study. And this is one of my this is one of the papers I read a lot in the Hao-Fountain field. So I wanted to highlight it also from her team if you would like to take a look into. And the next speaker was Dr. Manasa Proshottama from, again, Amsterdam University Medical Center. And she gave a talk titled "Decoding USB7 Dysfunction from Multi-omics Insights to Mechanistic Understanding." And they used patient fibroblasts, and they investigated multiple factors in this Hao-Fountain patients. And one of the factors was alterations of methylome, and most of the patients showed hypermethylated. Showed hypermethylation. And I think it was aligned with the previous study on epi signature profiling, that I think hypermethylation was reported more than hypomethylation. Then we had our first open discussion led by Dr. Clementina Fontasser at Texas Tech University. It was very informative, and it was between parents and scientists. Parents asked about the differences in episignature and why is that, and also is environmental are environmental factors affecting this episignature profiles of the patients. And we also discussed genetic testings and insurance. And Dr. Schoff gave us very good information about this genetic testing that if you do, for example, if you do whole exome sequencing, you can ask for your results for the raw data. You can get it from the clinic. And also the insurance, like how expensive it is, and some challenges that I'm pretty sure Becky and Bo have more information than me, like are some challenges with the insurance companies to that you can cover your genetic tests. And one of the parents, I think, brought this brought some information about dental issues, that I think her kid had more teeth than normal. And I think they are doing a study on that. And with this, I will leave it to, I'll tell one more slide and I'll leave it to Dennis to tell you the rest. It was Patrick's story. Patrick is an adult with Hao-Fountain syndrome, and he was there with his parents, and he shared some interesting part of his life. He is independent. He's living by himself, and he has a job. He works at a park, and he was diagnosed last year. And also, her parents printed out his parents, sorry, printed out some interesting information about him in case like we would like to know more. But talking to him and knowing that he's living by himself is fascinating. And Dennis.
Yeah, so our next presentation was by Dr. Christian Schaff from the University Clinic in Heidelberg, and he walked us through the more clinical viewpoint of USP7. He talked about the underlying mechanisms of the USP7 mutation. He also brought forward a very important piece of information, which is that they do keep registries of rare and ultra-rare neurological conditions with available biomaterials at Heidelberg. And in this specific registry, they also have 235 individuals with USP7 variants, which is, I think, 66% of all diagnosed patients. And they're also doing the first prospective study to investigate the metabolic, hormonal, brain, and skeletal aspects of Hao-Fountain syndrome. Yeah, they invited patients to Heidelberg, and then I think it was a 3-day study. And yeah, he also brought to our attention these two papers. The first paper to characterize the phenotypes of Hao-Fountain syndrome by Michael Fountain. And then this newer one with 32 novel patients by Moritz Wimmer. Then Claudia Esposito from RCCS Stella Maris talked about USP7 duplications. She opened up with Hao-Fountain deletions, that these deletions are not necessarily just the USP7 gene, but also can encompass a larger portion of the chromosome. I think the biggest ones are around 4.5 megabases. And she also pointed out that there are distinct facial features between patients that have a USP7 loss of function and a USP7 duplication. And the next story was a very, very inspiring story by Beth Smith. She talked about her life experiences living with Hao-Fountain syndrome. She was there with her husband. She talked about how she met him, her hobbies. I think she's a self-diagnosed adrenaline junkie. She does zip lining, I think. And yeah, she also talked about her parents' experiences about living with a Hao-Fountain child. Next was Isabella Jean-Maisonnet from Dr. Burlage's lab at the Dana-Farber Cancer Institute, and she talked about small molecule activators of USP7. They focus on developing these small molecules. She did a high throughput screen to identify a USP7 activator, and what she found was the MS8 compound, which the next speaker elaborated on, which was Dr. Emily Korchak from Dr. Besonova's lab at UConn. She investigated 11 mutations in the catalytic domain of USP7 and how these mutations affect the activity of USP7 and its binding to other proteins. And what she found was that MS8 enhances the activity of this mutated USP7. Next, we had a presentation from Charles River by Dr. David Fisher. He talked about Charles River's experience in drug repurposing and how they could apply this to Hao-Fountain syndrome. He opened up with explaining what they actually do, which is finding new purposes for these old molecules. These molecules are usually small molecules, either approved by the FDA or currently in clinical trials. He explained how their whole process works, how they choose the model to do the tests on, and then he provided some real-world examples of how they actually help people with very rare disorders. These are disorders I think only 1 or 2 patients were diagnosed with. He talked about Kendall with Lie syndrome, and he also talked about the Ryan Research Initiative. The next talk was from Dr. Janelle Stanton from Florida State Institute of Pediatric Rare Diseases. She talked about building upon the drug repurposing program. They are working on a drug repurposing screen for Hao-Fountain syndrome. In this screen, they're using a human colorectal cell line for the development of a high-content imaging assay. And through this assay, this is a microscopy technique, I think, they're trying to quantify changes in USP7 levels. And finally, we had presentations from our colleagues from Combined Brain. The first one was Elora Marvasi. She presented on a disease concept study. The next presentation was by Keelan Weston. She talked about the Hao-Fountain syndrome strategic plan. She talked about the timeline of the Hao-Fountain syndrome foundation from its inception to today. And she also pointed out what the Hao-Fountain community is doing good and where there are still some gaps or areas that need improving. And our final presentation was by Dr. Terry Joe. She is the mother of an Angelman patient, and she talked about her very inspiring story towards a PhD. And she also brought to our attention the need for social media and advertising to bring attention to these rare disease communities. That's all from our side. Thank you, everyone.
Thank you so much for that recap. Does anyone have any questions about anything that they mentioned? If you do, you can feel free to ask. And I think, if not, I think Kellen, you're up next, correct? Are you presenting today? Yes, right?
Yes, I was asked to present on the clinical presentations. So I can talk about that a little bit. So this will be somewhat redundant. Thank you, Dennis and Tara. So I'll be giving a little bit more of a recap of sort of clinically focused, but also trying to really talk about how each of these pieces of information, each of the highlights from the conference can really matter to the patient community. And so I'm going to focus mainly on, let's go this way, the 2 specific clinical presentations in the clinical section of the day, and then also the Combined Brain side. So we've kind of been over this, but that Hao-Fountain syndrome, Dr. Christian Schaff gave a wonderful presentation on the clinical phenotypes of Hao-Fountain. Everyone on this call probably knows of this study. I think what was really unique here was the development of this severity score for Hao-Fountain syndrome. It's a score like this that often can be very, very critical for future clinical trials for rare disorders like Hao-Fountain syndrome. And in his study, he shows that you actually have this genotype-phenotype correlation where you have a significant difference in severity score when variants are in the catalytic domain versus in other domains of the protein. And so it's scores like this that can actually help really spark more industry interest towards like looking for clinical trials. I really want to just highlight the registry by Dr. Schaff as well. The fact that this registry exists is truly amazing. And again, Combined Brain, we work with lots of rare disorders. And this is already above and beyond what so many disorders we work with have. And just knowing where these patients are, their contact info, and even the contact info of their physicians is critical. We also have been able to pull information from that. Dr. Schaff talked about how the inheritance, we have enough patients to really start looking at true percentages of inheritance, and how that for mosaic parents, we're able to conclude there's a 2% chance of having another child with USP7. So that's good to know, especially for the genetic counseling world. And also kind of the question of that started to get bandied about is how can we sort of take this amazing information in this registry and start to really connect it to the information in our Matrix digital registry that Becky talked about at the beginning of this call. Also just sort of some highlights of like what from Dr. Schaff's work was really, really piqued my interest and I think will be really important going forward for the Hao-Fountain community, Hao-Fountain syndrome community. It's really interesting that, or it's amazing that he collected CSF, blood, and photographs of people's faces. It has really just emphasizes the importance of patient-derived samples for study of Hao-Fountain syndrome. So the cerebral spinal fluid went to the Combined Brain Biobank, and a lot of the, we've got many, many more blood samples also being sent to collaborators that is sparking amazing research. I think at one point in the conference, we said, raise your hand if you're a scientist and you've used a patient sample from the biobank. And there was quite a show of hands. So just showing that patient sample donation matters and really is used. Again, Dr. Schaff contributed to the facial gestalt database, again, finding that Hao-Fountain syndrome has this distinct facial feature structure. That's really critical for diagnosis. Also showing that 91% presented with autistic features, but only 55% were actually diagnosed with autism, which really shows maybe there's a gap in diagnosis that maybe we could address as a community. Another interesting finding was that 25% of patients were on growth hormone therapy. And that one's important because that's very much underreported in the literature. It's also seen and used in Prader-Willi syndrome and Schaaf-Yang syndrome. And this is important. This is something that we should talk to the community about because hormone therapy, can help shift body fat to skeletal muscle mass. So it could really help with a lot of those developmental milestones, especially a lot of hypotonia issues or musculoskeletal issues. Dr. Schaff also pointed out that some individuals had glucose levels in the pre-diabetic range. That's important for the community because we can maybe tell parents or tell their clinicians to watch out for developing pre-diabetes or developing into diabetes. Scoliosis and bone health, having your doctor monitor bone health. This was a topic of discussion. Dr. Schaff at the end says that this is ongoing work of his, but that he is studying the kidneys and the urinary system. He highly recommended kidney ultrasounds, actually, which is something that I feel like you don't hear too often. If somebody has intellectual disability, kidney ultrasound is not the first thing that comes to mind. But this is the kind of thing that we can discover through studies like this. There's also kind of an open call of maybe somebody needs to look at kidneys and mice. So this is an opportunity for collaboration. So just again, emphasizing how each of the little findings can have, huge impacts on the community and how people's quality of life. Also, one thing that I am happy and over the moon about is that Dr. Schaff also has finished preparing a gene reviews article for Hao-Fountain syndrome. I don't think it's quite published yet. I just checked this morning to see if it's out. I didn't see it, but I think it's completed. And so Gene Reviews is this, for those who may not know, it's this online book that's just a big encyclopedia of genetic disorders. And Gene Reviews really is the go-to one-stop shop for researchers, clinicians, genetic counselors. Whenever I write up any report on genetic disorders, Gene Reviews is my first resource to go to. So having this will be very important, especially for clinicians, doctors, occupational therapists, physical therapists, really anyone who maybe is seeing a Hao-Fountain syndrome patient for the first time and has never heard of it before. Googles it, and this will be probably one of the first search results, and it will have just a wealth of information of everything that I just said on the previous slides and more to really help improve patient care. Also, Claudia Esposito, I'm kind of recapping this, but talked about dosage sensitivity of USP7. I thought this was just fascinating that there is this unique facial gestalt that they're finding. This actually can be really, really key for diagnosis in the future. And the fact that there was kind of this genotype-phenotype correlation, that's wonderful to see, and actually could even be like a future reference, again, used in clinical trials, where you have loss of function and duplication patients having different facial gestalts, with the duplication patients actually looking very similar to each other, while the loss of function patients have this much more variable facial gestalt. Next, I'll go over what Alora talked about with her disease concept model. So Alora was a genetic counseling master's student at Rutgers University, and this was her master's thesis, and she collaborated with Combined Brain to do this thesis. So for her disease concept model, it started with a literature review where she did an in-depth review of all of the clinical literature of Hao-Fountain syndrome, and then did qualitative interviews. This was, I think she did approximately 10 caregivers of people with Hao-Fountain syndrome, and did an open-ended interview that lasted approximately an hour and a half per caregiver, and just asking general questions about their quality of life, their symptoms, their day. What does a good day look like? What does a bad day look like? What are your primary impacts on your life? These open-ended questions that really allowed us to dig into getting a full picture of kind of what it's like to live with Hao-Fountain or with a person who has Hao-Fountain syndrome. We then took transcripts of those interviews and coded them, and then analyzed that data. And so here's just a graph of the symptoms that Alora found. And everything that has a star here was something that she found when comparing the published literature to what came out in the disease concept model, that these things were underreported in the published literature. So we saw expressive communication, speech, global developmental delay. These are all very well known. But things like frustration or abnormal crying or specifically speech impediment, these things were not as well reported in the literature. She also pulled a lot of quotes that she thought were really, I guess, exemplary of Hao-Fountain syndrome. The first one here, I think, describes really well sort of a lot of the behavioral issues that can be reported in Hao-Fountain syndrome. That maybe because of this lack of ability to communicate, Hao-Fountain patients can get very frustrated and sort of take out that aggression, and sort of talking about that. Alora also, in her talk, talked about how there was one, this is an example here in this quote, that schizophrenia was one thing that came up in one of her patients. And so that's maybe something that needs further investigation. So this was a patient who was having sort of schizophrenic hallucinations or delusions, and even had to be committed a few times into a psychiatric ward. And so is this something that's exemplary of Hao-Fountain syndrome, or that maybe, maybe your risk may be increased by having Hao-Fountain syndrome, or is this sort of a one-off case? And this may require further investigation. Alora's work also showed kind of the impacts of daily living with Hao-Fountain syndrome. So in this visual here, the larger the box, the more parents reported this as something that impacts their daily life. And so assistance with activities of daily living, so requiring support for just, daily living things such as, getting dressed in the morning, getting up, making food, all of these things was maybe the biggest impact. Dressing assistance, eating and drinking assistance, toileting assistance, bathing assistance, household tasks, mobility, adult day programs, or regression activities of daily living. These are things that were just most frequently reported. And information like this is important because, again, we can use this to sort of start to decide which types of meaningful changes can we look for when it comes to future therapies for Hao-Fountain syndrome. Often when it comes to therapy development and clinical trial design, there may be, I'm going to use, I know this is not as huge of an issue in Hao-Fountain syndrome, but I'm going to use seizures as an example, which is often used as one. Maybe, there was a 50% reduction in seizures reported as a result of a certain therapy. But maybe that doesn't actually impact people's lives as much as just being able to go to the bathroom independently, or that person being able to dress independently. If you actually ask the caregivers, if you could have one thing be, one thing, a therapy that could, magically cure one thing, parents often will report these activities of daily living over and above a lot of those, clinical reported things like expressive communication or, seizures, things like that. So this can be used to sort of guide how we measure improvement in Hao-Fountain syndrome as a result of therapy in a way that takes the actual lived experience into account. Here's just some highlights I thought from the Q&A and discussion that came out throughout. So these were just questions that parents had that I thought were really critical to keep in mind and continue to answer and dig into. Was the question of Hao-Fountain syndrome. Some parents wanted to know, will it remain rare? How rare will it be once we have, in a perfect world where we could genetically diagnose everybody, what would be the real prevalence of Hao-Fountain syndrome? I think that's actually still really a question that's in the air. How do we ensure more people get diagnosed? There were, like mentioned before, insurance questions. A lot of questions for how to be an advocate for your child, how to be a parent of your child, and help your child live their best life. There was a question, this was sparked by Boris's wonderful talk. What about infections and sicknesses? Do these children get infections more frequently or more severely or both? Is this something that is understudied, perhaps? Another question was, or things that parents reported helped were having IEPs, that's educational programs for their children. Exposure to new experiences, which is something that may not come first to the clinician's mind, but just, putting your child out there and helping them explore the world on their own, or even having special college programs. Some interesting phenotypes that came up that maybe haven't been reported in the literature as much was hair pulling or dental problems. So kind of going on to my and Terry Jo's portion, I'm kind of skipping around here a lot, was combined brain and sort of how we can help collaborate with the Hao-Fountain Syndrome Foundation to sort of build this, broad overview strategic research plan for Hao-Fountain syndrome. So we at Combined Brain show this slide a lot here, but here's kind of how we conceptualize everything from, a gene being ID'd as being causative of neurodevelopmental disorder, all the way to trying to get therapies, whether that's a gene therapy or a drug therapy for that disorder. Everything above the line here is really patient-focused data, with everything below the line being more basic science data. So sort of I do have a slide on this. Where is the Hao-Fountain syndrome community doing well? and then where are the gaps where the Hao-Fountain syndrome community should focus their efforts. I'm just showing, I guess, highlights from my slide deck. The first thing that I think is a wonderful gap that just got filled is the fact that, like Becky mentioned, we got an ICD-10 code for Hao-Fountain syndrome. It's Q87.87. It's just a simple little number, but this number is critical. I help a lot of disorders get codes. It can take years to get a code. This happens through the CDC. There's a certain committee at the CDC that decides these things. And this code can lead to much more accurate diagnosis for patients. It can lead to tracking healthcare outcomes for Hao-Fountain syndrome because this code can be pulled by maybe a pharmaceutical company, an industry company, pulled by researchers, where they can actually see where are the patients being seen, who are their clinicians, what kind of electronic health record data can we pull along with this, what kind of symptoms are they experiencing, what kind of surgeries or even interventions or medications are they taking. This can really look at Hao-Fountain syndrome over the course of the whole population. And it's really critical actually for getting pharmaceutical companies to actually become interested in your disorder or even start to think about investing money into developing a therapy for your disorder. Because once you have this code, you can find patients, and finding patients is critical for clinical trials. And then eventually, finding patients as far down the line, potential customers for a new therapeutic intervention. And unfortunately, in the United States of America, you can't actually talk about this to the CDC. They are literally not allowed to talk about this. But the real reason that a code will impact people's daily lives in a positive way is that it's used as an insurance billing code. So having this code, day to day will actually help parents get reimbursed for things like the dental issues, for things like, all of these symptoms that don't seem to go with a general like intellectual disability or developmental delay code. This specific code will really help people financially. We have natural history study on the matrix. I spent some time really talking about just how important a natural history study is. So the matrix, Becky already advertised at the beginning of this talk, but we have on the matrix, we have Hao-Fountain syndrome's genetic reports, consent form for biorepository sample donations, demographic data, surveys that can measure changes in symptoms over time. And that's really the critical part of a natural history study, is asking the question, how does Hao-Fountain syndrome change over time with age? What is the natural history, the natural progression of this disorder, which may not always be clear, especially when I think in Dr. Schaaf's registry, the average age was 14 years old. So we still have a relatively young population. So let's start asking what happens with age in Hao-Fountain syndrome. The matrix can pull in electronic health records. It takes quite literally 5 clicks of a button, and you can just auto import your electronic health record data. And there's also a clinician portal. There's kind of like a back end that we can hook up your clinician with, and your clinician can answer surveys or upload documents or see which surveys their patient has taken, which is just amazing. I think I may have already kind of verbally said a lot of this, but the reason natural history is so important is because this is solid data that can get researchers, can get industry, can get pharma companies interested in developing therapies for your disorder. It can answer questions like, where in the world are patients? Which types of variants should they focus on? Which symptoms can we target? Because which symptoms can we meaningfully measure changes in with surveys? Which age groups, which surveys. And it's also linked to the Combined Brain Biorepository. So we actually have this clinical information linked to the actual biological samples, which is just key. I also had shown this graph mostly for the parents in the room, basically as an advertisement to say, please sign up for the matrix. So if we have as of the conference, I have not updated this, there were 90 users in the matrix. So we can definitely continue to add more patients to the matrix. I also quickly discussed just models for Hao-Fountain syndrome and just how important models are, and how While there are many good models of Hao-Fountain syndrome, it seems to be the consensus that we could use better models. So how do we push our efforts into funding the creation of better models, or who might be best to create better models of Hao-Fountain syndrome? Also, just the value of these biological samples and the many different ways they can be used for research. And then kind of looking at, again, going back to the big picture. So we've got the gene ID'd, the disease concept model with Allura is mostly complete. We're working on natural history data collection. Mouse and cell and other animal models we've got many good ones. We can get some more better ones. Mechanisms, we had so many wonderful talks on really getting to the underlying molecular mechanisms of dysfunction as caused by these variants. We still need to look for better biomarkers of Hao-Fountain syndrome. That's something I didn't cover just now. But also, how can we make sure safety and efficacy is a thing for any potential small molecules or future gene therapies? We talked about the MS8 activator, something like that. And then in the future would be going towards clinical trials to try to find therapies for Hao-Fountain syndrome. And this is kind of just the big messy version, more like the back end version of this is the same timeline from left to right, but green is everything that has been initiated by the Hao-Fountain syndrome Foundation for Hao-Fountain syndrome, with yellow being in process, red are the gaps, and then blue are things that we'll need to put future investments into. So with that, I just want to say thank you to everyone, and yeah, it was a wonderful conference.
Thank you so much for your update, Kellen. That was fantastic going through all of that. I think it's such good information for all of us to have to know what our gaps are and, you know, where we're at as a whole. Also, at the beginning, when I first spoke, I said if anybody new is here, if they want to introduce themselves. I see there's a couple more people that came on after that. So if you want to introduce yourselves, you can feel free to right now if you'd like. For anybody.
My name is Jonathan Sabat. I'm a professor of psychiatry at UCSD.
Hi, thank you so much for joining us. And then I just have one last slide that I'm going to share. And this is kind of what we're going to be, you know, doing in the future for, you know, 2026 coming up. And that's publish the rare disease concept study, publish a strategic research plan, which is what Kellen was really talking about in her presentation, publish a standard of care, really get the matrix data analysis going, hopefully find some centers of excellence, places that, you know, more than one patient can go, that we can kind of focus on that. For scientific understanding the mechanisms and our small molecule screen, lab meetings. So for 2026, one model inventory wish list from you guys, like what kinds of models you would like to see. And then Jonathan is on the call. We are going to have him and Christian on for one of the lab meetings to kind of discuss the link possibly between USP7. I know his paper, I think it showed more about a duplication. But we do have, as Kellen mentioned, one definitely diagnosed. There's actually, I think, 2 more that are diagnosed that are young adults. And then there's 1 or 2 that have symptoms, but no formal diagnosis of schizophrenia. I don't know if they're deletions or if they're mutations. My guess is most of us in the group, it seems to be a lot of missense mutations that seem to be the biggest thing. So, you know, it'll be interesting and to hear from.
Do you guys have quantitative assays of USP7 function? Because my expectation would be that a fraction of the missense variants would be gain of function.
So
and would be different from Hao-Fountain syndrome.
So I think, and Emily, you might be able to answer that,
Yeah.
with the work in your lab.
Yeah, so we published a paper in PNAS last month, and we did 11 missense mutations in the catalytic domain of USB7. And when you just do the catalytic domain alone, some of them are hyperactive, but if you do the full-length protein, they're all less active than the wild type. So we're kind of like still lost as to how it functions fully.
Well, they may all be loss of function also.
Yeah, and it was just
Gain of
11.
function,
So
like
there's
they may
also
not be. I mean, it's not every protein can be. You can't produce a gain of function of every protein.
Yeah.
Channels. It's like if it were a channel, that might have a better chance, right? So the duplication may be the only gain of function we know about, right?
Yeah, and we do have a population of them that are part of our group that we include with our group. I think the presentation said there's 11 known of those people in the group. So that'll be something as well.
Something to keep in mind as one moves forward. In some of the other disorders that have been, published and known about for a couple of decades now, once the patient groups get large, they get a little bit disorganized and they tend to forget what they have and what subtype of the gene or disorder they are, and then they'll start giving each other advice. Oh, my kid looks like this. Oh, my kid looks like this, and then they have completely different disorders, and
Yeah.
So something that could be kind of applied early on is a good sort of like make sure that the advocacy groups are helping the families to understand this is what you have. It's different from this. Otherwise, they'll just get on Facebook and
absolutely.
yeah. But yeah,
And
it's just something to keep in mind.
I think even within
It's
our
something that we
small
see in
group-
other disorders that have been around for a long time.
Yeah, and even in our small group, it's such a variation of the spectrum from high functioning to, it's just such a wide patient group. So this is kind of what's coming up though for next year, the things that we're gonna try to talk about and move forward. Does anybody have any questions, or Bo, did you want to chime in on anything?
I think that about covers it. I think you got everything, Becky. Thanks.
I know. And I was like, when I first got here, I just had to go get Ella from school right before it started. So I was like, I was rushing because of course they called. So I was like, oh gosh.
No surprise, multitasking, right?
Yes. All right. Well, thank you all so much. And it was great to see everybody. And I think we're picking it up not again until the new year. All right. Bye.
Thanks, Becky.
Thank you. Bye.